Muy bien.
Si posso passar esse teclado aqui. Acho que tinha. Podíamos muito, não tem aqui. Mira, dos vezes. É um lac. Questo mi pare senza sortire.
So good morning, everybody. Thanks for being here. Sorry for this delay. We wanted to have everything ready this morning. So, well, some of you know me. I'm Eva from SOFIP. In SOFIP, we try to we work very closely with TMOS and rational oceanography, but also we expand to biogeochemistry and biology with different projects. For example, the turtles with telemetry that is led by Mark, or the bluefin tuna project with using operational oceanography with Diel. And we are very happy that today María hosted María Calleja. María Calleja, some of you may know her. She did the PhD here. She finished in 2007, so some of the faces may be familiar. And she is now located in the Max Planck Institute in Mainz, the pronunciation, Mainz, and in Germany. And she's interested in biogeochemical cycles, especially sulfur, nitrogen, and carbon. And she's here today to share with us her expertise. And she will talk today on carbon transport and the processes and links with organisms and vertebral migration. So thank you for being here.
Thank you. Yeah, well, thank you all for being here. As Eva said, I did my PhD here, so I can see some familiar faces, which is always nice. And I will start a little bit introducing myself, what has been my trajectory so far in broad terms. I will talk a little bit on some of the recent research activity that I have developed during the 4 years I spent in working in the Red Sea, mainly. And then a little bit where am I at now and future work ideas. So, as Eva mentioned, I did my PhD here. I actually started working as a technician in Carlos Duarte's lab, and I ended up doing the PhD. So I spent actually a total of 7 years here. And I'm also from Mallorca, so this is home from me. For me, and after during my PhD, I worked both in oceanographic and coastal processes that involved carbon and nitrogen cycling, and also a little bit of sulfur cycling in the coastal ocean here in the seagrass sediments. Then I moved to University of California in Santa Cruz, where I spent 2.5 years doing a postdoctoral stay, where I focused more on the chemical structure of the OM. So I used more my chemical expertise, which was my bachelor. And then I moved back home in Granada. I did another 3 years of postdoctoral stay in the Andalusian Institute of Earth Studies. then I had to leave Spain again. And I worked during 4 years in Saudi Arabia in the Red Sea, where we did both open ocean and also a lot of coastal work, both in seagrass systems and coral systems also. And now, since March 2019, I'm in the Max Planck Institute for Chemistry in Mainz. This is just an overview of the different oceanographic institutions I have been part of in orange, or I have been collaborating with in blue. So, and the big orange dots are places where I have done more intensive field work in coastal waters. So I was lucky enough to participate in different expeditions covering the tropical latitudes and the polar ecosystems too. So as an introduction, I will just start on talking a little bit on how I started my research during my PhD. We were looking at ARC CO2 fluxes, and I'm going to focus my talk here in oligotrophic oceans, even though I have also been doing some coastal work. So we were measuring CO2 fluxes between air and water, and what we wanted to check is whether not only in the coastal ocean but also in oligotrophic waters there was control of the fluxes due to metabolic processes occurring in the surface ocean. So we were measuring net community production rates in the very top centimeters of the ocean, comparing it to the net community production rates at 5 meters, which is considered surface ocean normally in oceanography. And what we found is that there was actually control on the PCO2, the partial pressure of CO2 in the surface ocean that was affecting the fluxes, but that control was happening in the top centimeters of the surface ocean. And of course, when there was net autotrophy, the ocean was acting as a CO2 sink. And when there was net heterotrophy, not always, but when heterotrophy was more extreme, it was also acting as it could act as a CO2 source to the atmosphere. So that changes seasonally and regionally, and yeah, that was actually a global study where we included a lot of different latitudes. But then, after the CO2 is absorbed by the ocean, the question is where does the carbon go? And of course, we have to keep in mind that there is the solubility pump, which is actually moving a lot of CO2, and it's explaining a lot of the CO2 and carbon transport in the ocean. But what I'm going to focus here is in the carbon that has been photosynthesized and converted into organic carbon, and where does this carbon go and how does it cycle in the ocean. And photosynthetically fixed CO2 transform into organic carbon in the form of marine biomass, which is the alive compartment of this organic carbon, but it also transforms into particulate organic carbon that is partly, when I talk about particulate organic carbon here, I talk about the abiotic fraction, and dissolved organic carbon. And when we look at, in terms of carbon reservoir, how important are these different fractions, what we can see is that actually most of the organic carbon that has been reduced from the CO2 into the ocean is in the form of dissolved organic carbon. And most of it is actually in the deep waters, in the mesopelagic and bathypelagic waters below the 200 meters depth. So this is a carbon compartment that is actually doesn't have a lot of credit. There are a lot of chemists working on DOC to get to know the structure and try to understand the cycling, but in terms of oceanography, it's also hard to introduce DOC into modeling, and it's sometimes people tend to forget about it, but it's actually a very, very important pool of carbon. So DOC is a key piece of the equation when we try to understand CO2 fluxes and where does the carbon go after it has been trapped, the organic carbon, after it has been trapped by the ocean. It's also a pool that is not a static pool, it's a very dynamic pool, especially in the epipelagic waters where most of the recycling is happening. But it can also sink to the deep ocean, and it keeps being remineralized by heterotrophic bacteria. So, because I don't know if you notice here, but the amount of DOC that we have in the deep sea is around. This is actually from the late 80s. This number is slightly bigger now, but so the numbers have changed a little bit, but the order of magnitude are the same. So this magnitude of carbon is of the same order of magnitude of CO2, the carbon that we have as CO2 in the atmosphere, which means small perturbations on DOC can really have big effects on the CO2 fluxes. And after this introduction, where I hope you a little bit understood what DOC is if you didn't know, I'm just going to introduce what has been my recent research activity. I have focused in trying to understand the DOC, how the DOC is reacting and cycling in the ocean, and to do so it's important to look at different branches of the subject. So one thing is that we need to try to know what's the composition of this pool. So DOC is a very heterogeneous and complex pool of a lot of different molecules, and it's difficult to isolate both the DOC without salt, actually, because of the low concentration in seawater when you compare it to the concentration of salt that you have there. And also it's hard to isolate different individual compounds to tackle biomarkers in terms of what's important for what processes. But there is a big community that is working on DOC structure, and yeah, we can get to know how many amino acids are there, what's the relative composition of those, even isotopic ratios of those. So there is a lot of hard chemistry working on that. But normally these structural analyses are focusing on very specific molecules, and it's hard to get the whole picture. And then recently there are more and more optical sensors that are getting outside in the market, and there is a lot that can be done actually just by following the optical characteristics of the DOC. So the sedum, which is the chromophoric dissolved organic matter, is the part of the dissolved organic matter that is able to absorb light. And the FDOM is the fluorescent dissolved organic matter, which is the fraction of that the fraction of the OM that not only absorb light, but after absorbing it is able to emit it. So considering the different wavelengths of excitation and emission of the pool, we can try to understand a little bit the characteristics of this DOM. So I believe that these are two different approaches that should work together in order to understand the whole structure of DOC. There's also another very important thing we have to understand, which is because, as I said, it's a very dynamic pool, and it has multiple interaction with the microbes in the ocean. is being produced during photosynthesis, during autotrophy by phytoplankton. While phytoplankton is growing, they are exudating DOC to the media, and this is the DOC that is actually very yummy or very labile for heterotrophic bacteria to feed on. But there are many other processes that are affecting the pool of DOC, such as the viral shunt, even grazing, and there are a lot of microbial interactions we still don't understand. That there's also a lot of effort put on that side in order to try to understand the cycling of DOC. And of course, we also need to know the physical transport, because DOC is just moving with the currents, right? So things like water stratification is affecting DOC circulation, different current, water masses, and this is also something we need to keep in mind when we try to understand reactivity and cycling of DOC. Another thing that is important so because of the high complexity of this pool, a way to simplify, which has been used for decades, is talking about the pool as different sub-pools, let's say, different compartments that are identified depending on how fast what's the turnover that they have in the water, what's the lifetime that they have. This is a typical vertical profile of DOC. So you have higher DOC concentrations in the surface where it's being produced, and then it decreases with that. Okay. The labile fraction is the fraction that is normally produced in the surface ocean by phytoplankton primary production, and this is the one that is quickly recycled by heterotrophic prokaryotes, so it actually stays in the water in the order of minutes to days. It's one of the hardest to understand because of its rapid recycling. Then there is the semi-labile pool, which goes a little bit deeper into the water column, and this is the fraction of the OM that is being microbes take longer to degrade. So it's also remineralized, it's also used by heterotrophic bacteria, but it takes longer. So it can stay in the water in the order of months to a year. This is the fraction of DOC that accumulates seasonally in the epipelagic waters. And then the refractory fraction is the one that stays in the ocean for longer time periods. It's actually the one that is composing most of this super huge pool of DOC that I showed in a previous slide. And you can find it in all the depths, and is the organic carbon storage of the sea. Okay, because it's not used by bacteria, so it's a carbon that none of the organisms use it, and it just stays, remains, sinks to the bottom, and yeah. So what I did in the recent years, working in the Red Sea, is we were sampling a station that we had in the central Red Sea. It was a mesopelagic station of about 700 meter depth. And we went there during 2 years, and we went sampling if the weather conditions permitted. We sampled every month, and we had vertical profiles every month of different parameters, not only DOC, to check on vertical distribution and the seasonality, both in the epipelagic and in the mesopelagic waters. Also, not every month, but during every season, we went there for a period of 24 hours to see changes occurred during due to Diel cycles. So by doing so, instead we were there and we were doing vertical profiles every 2 hours during 24 hours. This is a typical CTD profile of that particular station. A peculiarity here is the Red Sea is very saline and very warm sea. And you can see that here, so the gray line is temperature. You can see that the mesopelagic waters don't go below 22 degrees. So this is a very warm deep sea, right? So any processes occurring there, metabolic processes, are probably going to be enhanced because of this higher temperature compared to other mesopelagic ocean basins. Also, the salinity is higher than in other basins in the deep sea, so we have quite high dense waters. And in that particular station, we never saw an oxygen minimum zone, which is the red line. Once the oxygen was depleted, it stayed depleted during the whole mesopelagic. Okay, so that's kind of the DCM, which is the green line, was normally between 40 and 100 meters, and it was changing seasonally. This is the DOC vertical distribution that we found. Here you can see I grouped the data in winter, which is the blue points, spring in green, summer in red, and fall in orange. And one of the things that we could see by the DOC distribution, the vertical distribution first, is that there was quite a high variability of DOC concentration. This is DOC concentration with that. Compared to other oceans, because normally this is narrower, okay? So, okay, we still don't know why that is, but this is what we saw. And then we looked at what was happening in the epipelagic, upper 200 meters, and what was happening in the mesopelagic, between 200 and 700 meters. And we found an epipelagic seasonality. This is the data grouped by season, and we can see how DOC concentrations are increased during spring and summer. There was, which is probably because of higher temperatures and also higher stratification, so there is higher values in the epipelagic. The changes were not significant, but it was actually very, very interesting to see this seasonality, because it's not easy to see these changes in in oligotrophic oceans. We compared the amplitude of this change with other oligotrophic well-known stations that are also doing very long seasonal studies, such as BACs in the Atlantic and HOTS in the Pacific. And we found that the amplitude of our changes was slightly higher, but very similar. And this accumulation of DOC, what generates is, so the one that is not accumulating is the labile one. It's being produced and consumed, produced and consumed, produced and consumed. We don't see how it accumulates. And but the one that is accumulating, it has the potential to move towards deeper layers. And part of it is semi-labile DOC, which will further contribute to microbial respiration in deeper waters. And part of it is the refractory DOC, which will just sink and stay. The next step we did was we wanted to see, in order to study how much of this DOC was remineralizing and how was that affecting oxygen changes, we studied the relationship between DOC and upper and oxygen utilization. So upper and oxygen utilization, in case you are not familiar with it, you have to think of it as the inverse of the oxygen. So the lower the oxygen, the higher is the more it is utilized, the less oxygen you have. So here you have higher values because the oxygen is very depleted. Okay, this is the distribution that we found over time. You can see already some intrusion of DOC to deeper layers in the left diagram. A little bit of the same in the right diagram of the upper and oxygen utilization, but in order to really study what was the relationship between both parameters without the effect of depth, because they are both also having a very similar profile with depth, what we had to do was define different isopycnal layers in the water column, which are horizons or layers that have the same range of density, and study what was happening within each of these layers between the two parameters. And we identified 5 different layers. In the upper 100 meters, I grouped here the 3 upper layers in blue. And in none of them, I don't know if you can see that the correlation was positive, so there was an increase in upper and oxygen utilization with an increase in DOC, but it was not significant. And this is probably because a lot of processes are occurring in this layer. There's production, there's consumption, affecting both oxygen and DOC. There is also exchange of oxygen in the atmosphere here. And it's hard to see what's the net effect of the microbial utilization. So there was no significant correlation. But in the layer between mostly 100 and 200 meters, which is the pink one, which is the one between the 2 red dotted lines, so this one here, which is these are the density ranges, okay, and the approximate depth ranges. What we found was a significant relationship between DOC and the upper and oxygen utilization. Negative, and I will keep going on that later on. And there was no correlation at all in the main mesopelagic layer. So if we focus in that particular layer where the correlation was significant, this is the relationship between dissolved organic carbon concentrations and the upper and oxygen utilization. What we see is that there is a decrease in dissolved organic carbon while oxygen is being utilized. So the semi-labile fraction here that was accumulating and transporting downwards is being consumed by microbial respiration, mainly in between this, within this isopycnal layer. Okay, and then we can do some calculations. We know what would be the expected slope if 100% of oxygen consumption was due to DOC respiration, and comparing it with the slope that we found, we can calculate that 21% of changes in upper oxygen utilization in that isopycnal was due to DOC respiration. And this is something, this is a number that we can use to compare with other ocean basins, and it's actually quite similar to, a little higher, but very similar to calculations in the Mediterranean Sea, for example. When we looked at the deeper isopycnal layer, which is almost the whole mesopelagic, and this is the so this is actually the whole water column, okay? I just wanted to put you on perspective, and this is what's happening in the mesopelagic. And what we can see here is that there's a high variability of DOC concentrations, as we could see already in the vertical profile. But the upper and oxygen utilization, so we saw that the oxygen was very depleted almost all the time, and the upper and oxygen utilization is just very high and quite constant. okay? And we were not really expecting that. We were expecting to see a correlation here because we thought that the main processes down here were heterotrophic bacteria just respiring, but we didn't see that correlation. And then we tried to see, okay, what else is happening in the mesopelagic here that could give us insights on what an explanation for that. And because we were doing also these Diel cycles, during the Diel cycles, we were also acquiring acoustic data, and this is an example of what we saw, and that was very permanent. All the different Diel cycles that we did, we found this pattern. So I don't know if you're familiar with that, but this is basically an echogram where you can see Diel vertical migration. So this is what's happening from midnight to midnight. This is noon, and this is the vertical, the depth, okay? So from another study, we know that the Diel vertical migrations in the Red Sea are mainly due to skinny fish, skinny cheek lanternfish. So there are very few zooplankton migrating. And that more than 95% of the fish community of this kind is migrating every day. So this is a lot of carbon moving up and down every day that actually we know is happening in all the oceans, but we have no idea how to quantify this carbon. So we realized about that, and I just briefly, we believe that they do that, they have this behavior because they are escaping predation. So they prefer to be down in the mesopelagic and dark ocean during the daytime. During the daytime, yes, because it's dark down there during the day, and at nighttime they swim to the surface and they feed while it's dark, so they can avoid being seen by bigger fish. This is a theory, and it makes sense to me at least. So, and they can swim very, very fast. This is done in less than 20 minutes. They go from the surface down to 600 meters. So we thought, okay, maybe this could be affecting whatever is happening with the VOC down here. And we had a look on, this is data from the Diel cycles. Okay, so we have every 2 hours a CTD cast. This is the bacterial abundance, the empty circles are average day casts, and the black circles are average night casts, because we wanted to see if we could see a very clear difference between day and night. Okay. This is vertical profile of bacterial abundance. We didn't see any significant difference on the profile here between day and night. But when we check on the percentage of high nucleic acid bacteria, what we saw is that during the day, especially in the mesopelagic waters, so this is 300 liters, the percentage of high nucleic acid was significantly higher than the percentage of high nucleic acid bacteria during the day in the same waters, the same station, 24 hours there. So that was already like, wow, maybe the fish are, maybe there's an effect down there of the carbon that the fish are moving and providing, right? Because it's not only that they are moving, so they are feeding up there, where the phytoplankton is producing this fresh and labile material. They are eating up there and they are bringing it down. During the day, they are in that deep scattering layer, they are metabolizing all this material and they are defecating, they are probably have, I don't really know, but they probably have, they are exudating also material. So there is all this fresh material they just ate that is all of a sudden down there. So what we believe is that this fresh material, they are providing labile DOC to the deep scattering layer. And the vertical profile of DOC didn't show significant difference between day and night, but it looks slightly higher during the day. But when we look at, this is a property of chromophoric dissolved organic carbon. So we were looking at absorbance spectra of this DOM, and out of this absorbance spectra, you can calculate these slope ratios that are a proxy for dissolved organic matter size, whether you have higher size of molecule or a smaller size of molecule. And there is this theory about the size reactivity continuum model of DOM, which says that the labile material is of high molecular weight, and while the bacteria are consuming it, you have an accumulation of low molecular weight DOM. So what we can see here, especially in the mesopelagic, is that during the day, smaller slope ratios indicate higher molecular weight. Okay, so during the day we have an accumulation of high molecular weight compounds which are more labile, and during the night probably these compounds are being consumed, and during the night we have more proportion of low molecular weight compounds. So there was these two things that were indicating that this diel vertical migration could have an effect on the DOC composition, and affecting also the percentage of high nucleic acid bacteria. We moved another step forward, and what we did was not only have these in situ measurements, but we ran an experiment in the lab. So we took water from the surface during daytime, water from the surface and water from the deep scattering layer. We filtered the water through 0.8 microns to get rid of grazers and just have the DOC and the bacteria in the water, and we incubated the water in the lab at in situ temperature and light conditions. And we followed DOC concentrations and bacterial abundance growth, bacterial abundance with time. And we also calculate the bacterial size, the changes in size. And what we could see is that this is bacterial biovolume. So the size of the bacterial cells at the beginning of the experiment and at the end of the experiment. This is the surface and this is the fish layer. And what we saw is that these bacteria, of course this is an experiment, so these are potential cell sizes, but maybe it's not the real cell size, okay? So this is the cell size that we found from the in situ water that we sampled. This is the size at the beginning of the experiment. There was a slight reduction here, maybe they were stressed or something. And this is at the end of the experiment when they reached the plateau of growth. And what we saw is that the bacteria in the deep scattering layer had the potential to grow more than double the bacteria in the surface. And we were actually very, very surprised about that. Yeah. But that was an extra indication that fish could have an effect on bacterial growth down there. And also the growth efficiencies were significantly different from one, because of the different incised cells, from one layer to the other. And this is the little fish that is doing all these things. It's between 3 and 7 centimeter fish. You can see a little bit the scale of the ones, the majority of the ones we found with the pencil. In the Red Sea, it's mainly the skinny planterfish or Ventosema pterotum. This is a fish that is actually common in all the oceans. And this is Diel echograms from 2016 of different oceanographic basins. This is the North Atlantic, the West Pacific, the Indian Ocean, and the East Pacific. And what you can see is that these migrations are happening everywhere. The proportion of migrating fish, migrating animals, I should say, because in other ocean basins there might be a lot of zooplankton migrating too. So the proportion of migrating animals changes from one basin to another. For example, in the East Pacific, the lower right panel, it looks like more than 99% are migrating, right? So there is no, everything that was here goes down and then everything goes up again. On the contrary, for example, here in the West Pacific, there is a proportion of migrating fish that they stay in the mesopelagic layers, and they do not always go up and down. So there is the different proportion of the migrating fish might have an effect on the different carbon that they are transporting up and down. And this is also snapshot. So this is actually, yeah, just a snapshot of what they could measure during 24 hours in stations in these different ocean basins. There are very, very, very few echograms in open ocean, in the different oceans. And we know actually very little. The migration can change seasonally. There are some studies that say that it can also change with the moon cycles, because if they are actually affected by the amount of light, if they are trying to escape predation, maybe when there is full moon, they don't go to feed, or there are few animals that risk their lives because they can be seen better than when it's when you have a dark night, right? So a lot of different things we still don't know about these migrations. But there is this work. It's the result of the Malaspina expedition, where they were working with echograms, and they had acoustic data all along all the transects that they did, and they calculated that mesopelagic fish biomass could be underestimated by an order of magnitude, because normally these fish were counted by catches. So they had the trolling nets, and they could get the fish and count how many did they have here or there, and make an estimate of how much biomass of fish you have. But they put the camera in one of the trolling nets, and they realized that when the net was going down, all the fish were going away. So the amount of fish that they were catching was actually much less than the amount of fish that was there, fish or zooplankton. And so this is an estimation, the tenfold higher biomass, but this is also again something we don't really know. We should keep working on that. And yeah, so the take-home messages would be that the DOC is the largest bioreactive pool of carbon in the ocean. Its reactivity and cycling depends on its chemical nature, the interaction with the microorganisms, and the physical-chemical properties of the water column. So different density waters might have different transport of semi-labile carbon to the deep ocean. And the semi-labile DOC remineralization in the Red Sea contributes to 21% of oxygen consumption, especially, well, not especially, particularly in this lower epipelagic-upper mesopelagic layer, around 100 and 200 meters depth. And the DOC variability that we see in the mesopelagic may be connected to the active transport of labile DOC fraction by migrating fish that could be fueling heterotrophic prokaryotes in the deep waters of the Red Sea. And just something to point out is that if you remember, in the Red Sea we had this 22 degrees down there. So what we see there, we might not see in other oceans, but we don't know. The kind of experiments that we run, there hasn't been run in other ocean basins. So the question is this also happening elsewhere or not? And if that's the case, it could have effects on when calculating the carbon budgets in the oceans, which are especially not understood in the deep oceans. I will just now go briefly into what I'm doing now. I started in March in the Max Planck Institute for Chemistry in the Department of Climate Geochemistry in Mainz, in Germany. And they just built this sailing vessel. It's a 20-meter sailboat, and I look at it as I guess you all know the Tara oceans, and there's this. I think what they are trying to do, what we are trying to do, is kind of have a Tara oceans, but working more on geochemical studies and paleo proxies. So we are typically 6 persons on board, 3 scientists and 3 crew members. I'm one of the scientists, and 50% of the interior are laboratories. And the main goal is to sample contamination-free surface ocean and also lower atmosphere. We go down to 1,000 meters depth, but we try the next step would be to try to go down to 3,000, but this is a little critical because you have to work with the platform that you have and the winch that you have to operate, and it is not easy. We have to do everything on board. The 3 scientists, we don't have support of like the UTM in Spain or something like that. So it's actually very exhausting and hard, but it's also exciting. We are, the idea is to sample the Atlantic Ocean at different latitudes and marginal seas, and we might cross to the Eastern Pacific too. So far we have been working in the tropical Atlantic. The boat has 3 times of sampling, as I mentioned: continuous underway sampling of surface ocean, continuous atmospheric sampling of the lower atmospheric boundary layer, and discrete samplings of ocean vertical heterogeneity. This is here a tube where the atmospheric samples are going to. So we have 2 labs, the ocean lab and the atmospheric lab. So this tube is feeding the atmospheric lab instrument, and there is Teflon tube going through the keel that is being directed to the ocean lab, where the water is sampled for different things. These are all most of the measurements that we do in the surface ocean, atmosphere. I'm not going to go through that, but if you have any questions, you can ask me. And these are some preliminary results of surface ocean data of some of the sensors. This is actually from Lanzarote to Madeira, where we did a couple of cruises because we were just setting up everything. So we had to first make sure we could have good quality data before starting. And then once everything was ready, we moved forward to from Madeira to Azores, and then the boat needed to go back to Germany, so we turned towards Vigo, and then now the boat is in Bremenhaven because it needs some work outside the water. These are a little bit more of data from the sensors, and I'm not going to go into detail of that, but a lot of data is going to be generated, and one of the challenges is actually data managing and quality data and interpretation. So it's a lot of work at sea, but a lot of work in the office also. And these are just some nice images to finish with. We had the luck to have a professor that was a microscopist photographer, and he took these beautiful pictures from different planktonic organisms. There are some foraminifera where we extract some paleo proxies and others. And maybe that's not worth mention. I'm gonna finish here. Any questions? I will take. Thank you for listening.
Thanks, Maria, for this very interesting talk today and clarifying some aspects of this carbon transfer. And I'm sure that you will have questions.
I have some questions. Thank you. It's nice to see you here again. So
Thank you.
Well, this study with the fish is very nice and it's very exciting, but I have some problems in interpreting this data because, first of all, have you calculated the biomass of the bacteria that are in these waters and in comparison with the biomass that the fish is creating in the same water? Because, I mean, 10 to the 5 bacteria per milliliter, I mean, with several cubic meters you have already a big biomass. So comparing both might be also important. Because this high DNA cycle every day, this is very weird. So how do you interpret this? What is a high DNA for you or for the people who know about it?
The percentage of high nucleic acid bacteria is taken from the cytograms.
I know. But this must be either DNA duplication, so it might be either chromosome duplication or might be ribosomal increase in the cell. So, but those are completely different metabolisms.
Yeah. We don't have the data to know whether it's one or the other, actually. We just have this observation and we actually were not expecting that. So it was coming all out of the data that we were getting. But I can show you.
But this is very important because it is not the same thing when a microorganism decides to divide itself or when he increases the level of ribosomes because the conditions might need more ribosomes to synthesize proteins.
Yeah.
So this is two different strategies. And I want to link this, and I will finish with this, with the volume that you saw. It is also very remarkable. So a microorganism only increases volume if it doesn't divide. So if you divide actively, very much actively, then normally you have small cells, but many cells. And if you don't divide actively, because sometimes you increase the length of the cell, because normally these biovolumes that you calculate are not because bacteria do not increase in volume. So a coccyte is always the same size, but only the volume increases because of the length of the organism. So, and to finish, normally organisms that they do not divide, they increase the levels of ribosomes. And the ones that divide actively, they have much more DNA instead. This is.
Okay. I have to say I'm not an expert in the microbial side, but what I believe is happening, and that might or may not answer your question, is that because the fish may be providing a source of more labile DOC down there, when they take up DOC, they do both. They use part of it to incorporate into their biomass and part of it to respire, right? And I believe that because they are getting more labile DOC, more yummy or fresh DOC coming up from the surface and excreted by the fish, they are using this DOC to incorporate it into their biomass, and that's why they are getting so big. And
they cannot be bigger. They will be big if they grow in length, but they don't grow in volume like animals, plants, or anything.
Well, because the calculation of the biovolume, it depends on the side scatter. It's an estimate of the side scatter that you get from.
The only thing that could be is that they accumulate some, for example, polysaccharides outside the cell, or things that they make the cell volume bigger because they accumulate extracellular material. This could be some organisms. They create a capsule around themselves, but the size of a bacterium is the size of a bacterium, so they don't get fatter or thinner depending on the conditions.
But that's
so the
what
volume
we
you
saw.
have to be careful with this volume stuff.
Yeah, and that's why I mentioned this is a potential growth, because that was happening in the incubations we had. It was not. These are not the volumes we find in the water.
Did you also calculate the number of cells in the
Uh,
experiment?
Yes, but I don't have it. I don't know it by
Because
heart.
this might be important to link,
Mm.
because you will know whether they are dividing or not.
To having a look at
Yeah,
both.
because the strategy
Yeah.
will be this: either I grow in length and I don't divide, or I actively divide and then we are much more. But because at the end the biome is the same.
Yeah, actually we could redesign another experiment and have a closer look to all that.
I think you should.
Yeah, thank you.
I also miss quality of organic, because it's not the same. You say very labile or not labile, but this will depend on the length of the compound. So whether they are sugars, whether they are not. So I miss a little bit of this chemistry, because you know it, but.
Yeah, because we were actually working with the optical characteristics of this DOM and not so much, we were not doing structural studies. We didn't have the instruments to do so. So all the DOC composition and this interpretation on labile or not labile was depending on the data that we had from the spectral slopes, which was coming from CDOM data. So I don't have structural information to tell you that. But normally the more labile material in the FDOM data, you can see higher signal of protein-like material compared to humic-like material. But that is coming from the proteins that are able to emit light within the DOM, which are tyrosine.
In my very last So imagine that a fish that is down there defecates. That probably does. Defecating probably is releasing, let's say, 100 microliters of feces, or let's say 100 microliters of feces, which will include around 10 to the 8 bacteria. Well, these 10 to the 8 bacteria that are just released there are probably high DNA containing, because normally in the gut they are much more active than in the waters. Do you think that this bacteria can be the one that you detect of high DNA after all these fish being down defecating and going up?
Yeah, I believe these are the bacteria that are responding to this defecation actually.
This would be
or metabolized products from the fish.
Because for me would be the only explanation to see this daily cycle of high low DNA. There is no other explanation.
Yeah, I believe that's actually the reason. Yeah, and also the high temperatures that might make a difference on other deep waters. But yeah, I believe that's the explanation.
Yeah, because temperature doesn't change. It stays the same down there. So it's stable.
Yeah,
so
I mean when you compare it with other deep ocean basins. But no, temperature doesn't change during the day, of course. It doesn't. Thank you. We should do an experiment together.
Yes. But if the fish can be eaten, if not?
One of the problems, we actually wanted to catch fish and to incubate fish and to get the dissolved organic matter released by this fish. But that was impossible because to capture a live fish to do that, we couldn't do it. It was when you take them from the deep ocean, once they are up there, they are almost dying, and to get them from the surface, they are in the surface at night. So trawling was not an option because they also were getting almost dead when they were up. And then we tried to dive and catch them, but we didn't get the permits to do so because it was night, it was open ocean. So we are correlating the two data sets, but we don't have actually DOC excreted by the fish. That would be ideal, and then feed the bacteria with that.
If you take a sample and you look at the gut fish microbiome, and you look at the sample during the day, and now you will see whether it is related. Because the composition of bacteria inside the gut is completely different than in the pelagic. And my only explanation is this: they are just out of the gut, and they start to die because they are aerobic conditions. They are not used to aerobic conditions. And
Yeah. And we were just looking at free living bacteria. We didn't check on the but that would be really interesting to do. Yeah.
Take the interest.
Yeah.
Shit. More shit.
Yeah. That's true. Yeah.
I believe you have more
Yes.
questions.
Thank you for your talk. And I don't know if I have missed it, but so the different kinds of dog has to do with their structure. And then I would like to know if it's possible to change from one to another, like from labile to semi-labile or from semi-labile.
Yeah, totally. So there is actually the heterotrophic bacteria not only incorporate DOC, they also transform it. So there are a bunch of reactions, both biotic, and the biotic ones are normally by heterotrophic bacteria remineralization. Sometimes the remineralization doesn't go from DOC all the way to CO2, but from DOC to other kind of DOC. That's also actually why the high molecular weight DOC is more labile, and it's been higher number of molecular weight DOC is being produced because there is this transformation that is done by bacteria, not only by bacteria but also by photochemical processes in the surface ocean. There is a lot of photobleaching that is also affecting DOC. So there can be oxidation of DOC from one form to another.
I think Maria is going to stay a little bit around anyway, so if anybody wants to talk with her, we'll have an opportunity. Thank you again.
Thank you.
Maria. Thanks so much.
Son molt malo. Però...
Ya os sabía que ahora. Caña.
Hombre...
No, pero si me entro ahora. Además, no, sacamos de i ells microbiòlegs i ells tot aquesta part. No, això és tot aquesta feina hem començat a estudiar. Ara que també m'he recordat el que i has d'entendre
reflexió.
Sí.
Això no molt xulo, en el mar negre, i vam veure que a 8 graus, per tant, sediment s'ubicaven molt efectivament, perquè tenen sempre molt